Hi students, this is A. Mandari Kumar Rajesh. In this video, I would like to teach you a new chapter called Chromosomal Basis of Inheritance. Let us discuss the topic Chromosomal Theory of Inheritance in detail. Mendel held that the traits were transmitted from one generation to another generation as discrete, stable, particulate units of heredity called factors. Nowadays, factors are considered as genes. According to Mendel, the factors are responsible for the transmission of characters from one generation to another generation, and he published his research work on inheritance of well-defined characters of pea plant in 1865 in the Proceedings of Bern Natural Science Society, but it was unrecognized for several years due to several reasons. So, in 1865, Mendel published his research work on inheritance of pea plants. Inheritance of pea plants. And it was unrecognized for several years due to several reasons. I give you some of the reasons why it was unrecognized. The first reason was the publishers failed to propaganda Mendel's work because publishers circulate limited number of proceedings around the world. That was the first reason. The second reason was Mendel findings were ahead of his time. That means his findings was very advanced at that time because there was no awareness about meiosis, mitosis, that is cell division, and there was no development in the microscopy. So his findings were advanced at that time. Then the third reason was generally Mendel was a mathematician. So his mathematical approach to solve biological phenomenon was completely alien for his contemporaries. So that was the third reason. And final foremost reason was at the time the scientific world was being shocked by Darwin's theory of evolution. So these were some of the reasons why Mendel work was unrecognized for several years. In 1900, Mendel's principles were rediscovered by the three scientists, namely, so in 1900, Mendelian principles were rediscovered by the three scientists, De Vries, Tessermark, and Correns. Then the researchers started to research on the unit of heredity, soon led to the development of chromosomal theory of inheritance. In 1902, the chromosomal theory of inheritance was proposed by W.S. Sutton and Boveri. T. Boveri. So, the chromosomal theory of inheritance was proposed by W.S. Sutton and T. Boveri in the year 1902. And in 1933, the chromosomal theory of inheritance was confirmed by T.H. Morgan. Then let us discuss the salient features of chromosomal theory of inheritance. The link between the one generation and the next generation is through the male gamete and female gamete, which carries the hereditary traits, because one set of hereditary traits received from the male parent and other set of hereditary traits received from the female parent. These two chromosomes constitute a pair of homologous chromosomes. I will explain this point with this diagram. So here we have two individuals. One individual represents the male, the other individual represents the female. Then during gamete formation, each chromosome segregate and form into gametes. So one gamete come from the male parent and which contains the hereditary traits of male, and other gamete come from the female parent which also contains the hereditary traits. So the hereditary traits are nothing but genes. And fusion of gametes takes place, results in the young one set of hereditary trait come from the male and one set of hereditary trait come from the female and constitute to form a pair of homologous chromosome. Now you get somewhat clarification. Then the chromosome retains their structural and individuality throughout the life cycle until it is subjected to a mutation. Then the third point is each chromosome carries a specific determinants. Specific determinants are nothing but the Mendelian factors. Mendelian factors are otherwise called as genes. So each chromosome carries a specific determinants or Mendelian factors. Now it is said to be genes. Then the fourth point is, according to this theory, chromosomes are the carriers of hereditary information which possess Mendelian factor, and that Mendelian factor can segregate and assort independently. Then they transmit the characters from one generation to the next generation. These are the 4 points which are mentioned in our book. Now I give you the similarity between the Mendelian factor and chromosome behavior. Here we have 3 points. The first point is alleles of a factor occurs in pair. Here the chromosome occurs in pair. For example, the capital A is an allele which is responsible for the tall character and they are occurs in pair. Suppose it may be capital A, small a or small a, small a. So they are occurs in pair. Here also the chromosomes occurs in pair, that is called homologous chromosomes. And in the chromosome, the alleles are fixed in the position called locus. So here also alleles are present, but they are present in the chromosome. That is the major difference between Mendelian factor and chromosome behavior. In Mendelian factor, Mendel said simply a factor is responsible for a trait, but in the chromosomal behavior they represent with a chromosome. In the chromosome the alleles are present, that alleles are responsible for the respective traits. Then the second point is similar or dissimilar alleles of a factor separate during the gamete formation. Here the homologous chromosome separate during meiosis. So during gamete formation the alleles get separated and developed into gametes. Likewise in the chromosome also during meiosis the spindle fibers separate these two chromosomes. So the two chromosomes developed into two gametes. Then the third point, Mendelian factors can assort independently. The paired chromosome can separate independently during meiosis. So please consider this is a dihybrid cross. So here we have capital A, small a. So that the A allele is responsible for the trait tall. So here we have capital B, small b. So capital A can assort independently with small b. This capital B can assort independently with small A. Likewise, in the chromosome also takes place. But in the condition like linked genes in the same chromosome, the independent assortment is not possible because it is a capital A, the capital B is closely arranged so that genes are said to be linked genes. These two genes are said to be linked genes. In that condition, that chromosome cannot assort independently. That's all about today's video. I hope that information will helpful for you. If you have any doubt, please clarify with me. Please stay home, stay safe. Thank you very much for watching this video. Thank you.